Regeneration is a process that is seen all throughout the animal kingdom, all the way from jellyfish up to salamanders. However, it is also known there are a lot of organisms that cannot regenerate or have very limited regenerative capacity, which includes most mammals and also humans. And a lot of research is trying to understand not only why organisms cannot regenerate, but also kind of putting together all of the information about organisms that can regenerate to get a better understanding about how we can activate regeneration in non-regenerative species. So in our lab, we use echinoderms as our model system. Now, echinoderms are a phylum of marine invertebrates, which include many different animals such as sea urchins, starfish, and sea cucumbers. And what's great about echinoderms is that they are amenable for studying of development, but also they're well known to be able to regenerate, including the adult forms, which if you cut off a starfish arm, it is able to grow a whole new body. However, as indirect developers, they have a larval form which also can regenerate. And so my specific angle on this project is looking at how cell proliferation plays a role during regeneration in this species, because cell proliferation has been shown in many different species to be important for regeneration, as you need new cells in order to form tissues that were lost. And specifically, I'm trying to understand where the cells come from that form the new tissues, where do they end up, and also how is this process regulated, and how is this different from proliferation that might be involved in just maintaining normal homeostasis. So another great advantage of using echinoderms to study regeneration is that their larvae are transparent, so we can do a lot of microscopy, including fluorescence and confocal microscopy. And I've had the great fortune of using the imaging center, EMBIC, in order to study not only proliferation, but also the effect of certain perturbation agents on proliferation and see how that affects regeneration. So the proliferation that I find is specifically turned on during regeneration involves a clustering of cells near the cut site, which is indicative of a structure that is shown in many different species, suggesting that this is a structure that is very conserved among regenerative species. And also this clustering of cells, the proliferation cells there, is very distinct from the cells that are proliferating not only in the normal larvae, but also early in regeneration, suggesting that proliferation during regeneration is specifically induced for the purpose of regeneration, and that the normal proliferation early in regeneration is turned off. So ultimately what we want to identify is not only the signal that is inducing proliferation specifically during regeneration, but also what is the gene regulatory network that is governing the pathway from proliferation to ultimate differentiation to these cell types that need to be formed in order for the larvae to form a full, complete patterned organism. So ultimately, this research is contributing to the growing knowledge about how regeneration is occurring in various different regenerative species, and echinoderms serve as a great model as they're very closely related to vertebrates as sister taxa, so we can compare regeneration in a species that is relatively closely related but can also regenerate very well.